Okay. GPCR, that is G protein coupled receptor pathway. So this is first thing we have to remember is it is serpentine, and second thing it is heptameric. So both of these terms means that there are seven main membrane spanning regions like this way: 1, 2, 3, 4, 5, 6, and 7. So this is how the complete structure looks like, and it is a alpha helix structure. So because we have studied about the protein structure in the unit number 1, so you must be knowing about that what alpha helix structure is all about. So as you can see in the figure, we have the plasma membrane. So this is the plasma membrane. This is the plasma membrane. We have the extracellular region and the intracellular region. So in the extracellular region, we have the N terminal, and on the intracellular, we have the C region. I'm going a bit slow in this because I want to make it very, very clear. So in the extracellular, we have N, and intracellular, we have C terminal or the C region. Okay. So now from the N terminal starts the very first loop. Then we have second loop, third loop, fourth, fifth, sixth, and seventh. So on the ending part of the seventh loop, we have the C terminal. So we have loop 1, loop 2, and loop 3. Loop 3 is a longer loop. Loop 1 is made up of 1 and 2. Loop 2 is made up of 3 and 4. And loop 3 is made up of 5 and 6. You have to remember about these structures because exam ke andar ye aata hai. That's why just remember about this. And we have the transmembrane structures as well, in which we have T1, T2, and T3. So we have transmembrane 1, which is made up of loop second and third. It is different from the loop 1, which is made up of 1 and 2. Transmembrane wala 2 se start hoga. Transmembrane 1 would start from 2, and the loop would actually start from the first one. So loop 1 is made up of 1 and 2. Transmembrane 1 is made up of 2 and 3. Transmembrane 2 is made up of 4 and 5, and transmembrane 3 is made up of 6 and 7. Okay, so this is the complete structure. In addition, loop 3, which is a longer loop, it is attached to this particular G protein. Now this G protein have 3 subunits, which is alpha, beta, and gamma. They are organized in this particular fashion. Okay, so what happens is loop 3, that means loop number 3, which is made up of 5 and 6, it transduces the signal to the alpha subunit. So when in the initial or the very first step, there occurs the extracellular domain, that is this one. The N terminal receives the ligand, and the ligand comes and bind to the N terminal. Now it would pass on from loop 1 to 2 to 3. So from the loop number 3, the signal would be passed on to the alpha subunit, that is this. So in G protein, we have alpha, beta, and gamma. We have alpha, beta, and gamma. The very first subunit which would take up the signal is the alpha. Okay. So in this particular thing, the alpha would take up the complete signal. Now you can see the ligand binds to the T3 loop, that is 6 and 7, or we can say the transmembrane 7 domain. Loop number 3, that is L3, 5 and 6, it transduces the signal to the alpha subunit. This is what you have to remember. Now we'll understand the complete functioning of the same. So just look at this particular diagram, and that is we have this pathway. In this, we have the N terminal and the C terminal. This C terminal passes on or transduces the signal to the alpha subunit, because of which the GDP gets converted into GTP. Now you can see we have alpha subunit, G alpha. ये clearly visible है सबको? इसको मैं थोड़ा और zoom करूं?
It's visible.
Okay. Okay. So we have alpha, G alpha, and GTP. Now what happens? there occurs जो ये G alpha और GTP है, it converts the ATP into CA. This ATP activates the CAMP. Now 4-CAMP gets activated. In addition to this, what happens is adenylate cyclase जो है वो यहाँ पर it functions in this way. Now 4-CAMP from the inactive state of pKa, they convert or they activates it into pKa active. Now this pKa active from the nuclear core, passing through the nuclear core, enters the nucleus. Now pKa activates CREB. So CREB is camp response element binding protein, which was inactive. It gets converted into the active form. Now CREB active along with CEB. CEB is CREB binding protein. They both combine together, and then the transcription gets on, and finally the response occurs. So on this CREB, after getting completely activated, it transcribes the target gene, and finally the transcription is on and the response takes place. So this is the complete pathway of the GPCR. We'll go through this particular slide as well. And that is the working of the GPCR. The GPCR binds to the ligand and activates a membrane protein called the G protein. Now, because we have understood about the complete pathway, you will get or understand all these points. Now, activated G protein interacts with the enzyme in the membrane, that is adenylate cyclase. Now, once the G protein gets activated, it changes its shape, it becomes active, and it splits into two different subunits. So as we have just seen, when the G protein gets activated, that means G alpha gets detached from it, that is the active form. And in this format, initially when we had three subunits, alpha, beta, and gamma, now the alpha unit gets distorted. It gets to, or it splits from the official or the original shape. Now after getting activated, it splits into two